Hello my dear student, I am Professor Devashish Bose, Head Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. Today I am going to present a lecture of BSc 3rd semester on the unit Criminalistics, and the topic will be sample handling. This lecture has been jointly prepared by myself and Mr. Giriraj Sharma, a PhD scholar and a UGC JRF at Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. So let's start our discussion by taking a look at what we are going to learn today. In today's lecture, the module 1 will be dedicated to Introduction to Toxicological Evidences. Module 2 will be Sample Handling in Forensic Toxicology. Module 3 will be Sample Selection and Number of Samples. Module 4 will be Sample Collection. Module 5 will be Sample Container, Collection Site and Sample Quantity. Module 6 will be Sample Labeling and Sample Preservation. Finally, the Module 7 will be our conclusion. So, my dear student, let's start our lecture with Module 1, that is Introduction to Toxicological Evidences. So, I think by this time you are well versed with the term forensic science, and you know that how samples plays an important role in solving a crime. That is, I am talking about physical evidences, and physical evidences contain sample. So, again the success story of our forensic science depends on the way we collect the sample. What are the samples? How to collect them? How to preserve them? How to send them? In this sequence, we are starting our lecture. So, we will be studying about the toxicological evidences. So, before starting toxicological evidences, we will be taking a look about forensic toxicology. So, you should know, it's a brushing of your memory that what is toxicology. So, let's rebrush our memory. Toxicology, what does mean the word toxicology? It may have roots from the stem of Indo-European word, that is tvek, T-E-K-W, meaning to flee, or may have derived from the Greek word toxon, bow, and the Latin word toxicum. So, these are the things which from where the toxicology came into existence. There are many history that where it got its origin, we found from where it's coming. So, there are a lot of people, lot of things, but it's a general idea. Thus, many definitions of toxicology have been proposed also. But in short, we can say that toxicology is the study of the adverse effect produced by the chemicals and drugs, or in other words, we can say that toxicology is the study of the adverse effect of chemicals, physical or biological agents on living organism and the ecosystem, including the prevention and amelioration of such diverse effect. So, this is toxicology. So, again, in a layman's language, how we can define it? It was a technical thing. Now, for a layman, how you can make a layman understand what is toxicology? Suppose you are moving in a street and talking about toxicology. You have to explain that person what is toxicology. So, as per him, toxicology is the study of poison and their effect on living organism. Simple. So, now again a new word came, poison. So, he will be asking you what is a poison. So, if we go to the history, because always we start with the history and come to the present. If we talk about the history, it is the same as the French word for magic potion, which take its terms from the Latin word potere or potre, that means to drink. Moreover, as Berzelius defined poison as everything is poison, there is nothing which is not a poison. This is only the doses that make it either a poison or a remedy. So, what he wanted to say is that everything which is around us are poison in nature, but who decides whether it is a poison or non-poisonous thing? It's the amount, it's the quantity. You can think over it. You can think over it and come back. So, here we are studying forensic toxicology. As we know that forensic toxicology is the application of toxicology to criminal justice system. At present, in the year 2006, Society of Forensic Toxicologists, that is SOFT, considered 3 branches of forensic toxicology. The first one is Post-Mortem Forensic Toxicology. This branch of forensic toxicology deals with the viscera sample, in which forensic toxicologist determines the absence or presence of poison that may be in the form of drugs or their metabolites, chemicals, plant and animal poison or other toxic chemicals. Forensic toxicologist evaluates their role as a determinant of contributory factor in the cause and manner of death. Next is Human Performance Forensic Toxicology. It includes the determination of the absence or presence of the ethanol or other drugs, chemical in blood, breath or other specimens, and they evaluate the role it modify human performance or behavior. The third is Forensic Urine Drug Testing. It mainly deals with the determination of absence or presence of drugs and their metabolites in urine to demonstrate prior use or abuse of some drugs or some chemical. Moreover, it also deals with the determination of the drugs and chemicals in the human hair, oral fluid, etc., etc. So this is how the SOFT, this is the main organization or association dealing with the toxicology all over the world. They see the classification for forensic toxicologist. This is it. I think it is clear. Just go a little bit into it and it will be clear. So my dear student, now we will be reading about the toxicological evidence. So when death occurs due to poison or overdose of some chemical, then the evidence would be accounted as toxicological evidence. For the sake of convenience, we can divide this category into 3 parts. That is, Ante-Mortem Samples. Means all those samples that from a person who has taken it or the person who has been affected by that poison is alive and we need to detect it. So it is known as Ante-Mortem Samples. It includes the following. They are blood, urine, vomit, nail clippings, hair, unwashed cloth, soil, empty containers, all these things, stomach wash, all these things. That means because he is alive. Now, next is post-mortem samples. That means if after the consumption of the poison, that person dies, then what samples are required by a toxicologist? The samples required are the viscera, the main important thing, viscera. They are liver, stomach, small intestine, kidney, spleen, lungs, heart, brain, bone, skeleton, skin, apart from all the samples which we can get. Now we move on to another, that is non-human samples. This category of toxicological evidences work in the absence of the post-mortem samples, like suppose the person vanished, they have cremated it or they have buried that person under the soil and it has been decomposed, then what you need? We need insects, if still there, soil, and the household and the personal samples. So I think now it's clear to you that what kind of samples we require as a toxicologist. With this, we come to the end of module 1. Now we start with our new module, module 2, sample handling in forensic toxicology. So my dear student, now you know about the samples. Now how to handle them is very important. If you don't handle them properly, suppose you get an alcohol sample and you transport it in open, or acid, you put that acid in a plastic container. So it's all spoiled. So now my dear student, you have to know that, okay, we know about the samples. Now what is sample handling? So sample handling is very important in forensic toxicology. As we know that sample handling is the most important part of the analysis. Without performing this part in a very well and a calculated, precise manner, the whole analysis may get spoiled and will be of no use. So extreme care should be exercised during the time of the sample handling, whether it is at a scene of crime or while taking reference sample from the victim suspect or perpetrator. So, first of all, we will try to understand the concept of sample handling. One thing is that you have to remember is sample handling is a systematic and step-by-step procedure. You have to prioritize your identification, individualization, detection and quantity of the analyte in the matrix, and obviously the matrix also. The important procedure of sample handling must take place systematically. Sample handling includes sample collection, sample selection, storage, preservation and finally the transportation. For the sake of simplicity, we use the term sample handling instead of many. There are many other terms. So, all these steps collectively will be referred as sample handling. Sample handling has great importance for the qualitative and quantitative analysis of the analyte. Moreover, if you want to get accurate and precise result, you need to make proper sample handling. Thus, proper sample handling will start the foundation or set the foundation for the accuracy of all analysis. Any error in these procedures may render all subsequent analysis zero. Here, I would like to clear one thing, that is why sample handling is so important to forensic toxicologists as well as analysts. It is important because as we know that concentration of the analyte in the matrix is very important. When this concentration alters due to mishandling of the sample, it would render the whole analysis useless. Your all efforts will go in vain. So, not only forensic toxicologists but also other who are going to collect sample must be trained in the correct method of sample handling. As we know that forensic toxicologists are commonly called upon to determine the presence or absence of the poison in the viscera, whether it is volatile, non-volatile organic poisons, insecticide, metallic poison, toxic anions or cations and other poisons. Therefore, an appropriate sample handling protocol must be followed by a forensic toxicologist for each and every type of sample. Moreover, forensic toxicologists must maintain the legal requirement such as the maintenance of the chain of custody. So, with this, we come to the end of module 2. My dear student, now we start with our next new module, module 3, sample selection and number of samples. So, now we will learn about the selection of sample. So, before jumping on to the sample selection, we have to understand the criteria for sample selection. As we know that selection of the appropriate sample is of most important nature, a step in the analytical process. In addition to this, there are several criteria that should be considered in the selection of samples, such as purpose of the analysis, if it is an ante-mortem case or a post-mortem case, sample availability, condition of the sample, type of interpretation, ease of analysis, drug stability. We will have introductory part about what we are going to do at the higher level. When you will go to the higher level, you will have more detailed study. But here, for the sake of convenience, we will going into brief. Hence, in sample selection, purpose of the analysis is most crucial step. It must be clear cut what toxicologist does with this analyte. For example, suppose that forensic toxicologists want to establish a fact that a person is subjected to drug or been exposed to a chemical. So, according to the analysis, one will collect the samples whether the analyst wants to perform the preliminary examination or confirmatory examination. While performing the analysis, forensic toxicologists should not go through the tedious method for the analysis. One should use the most preferable method of analysis. Here, I would like to clear one more thing, that the analysis is of two types. That is, one is qualitative analysis and the second is quantitative analysis. Now, I would like to explain you what is qualitative analysis and what is quantitative analysis. Qualitative analysis is to know what that analyte, what that compound, what that chemical is. Quality, we say na quality, अच्छा है, बुरा है. This is the quality. Whether it is of good quality, bad quality. Quality means we know about what is it. And next is quantity. It's a self-explanatory word, quantity, which means matra, which means the amount. So in forensic toxicology, it is very important, the quality and the quantity, what it is and in what much amount it is. So sample collection in forensic toxicology should keep in mind that what one is going to do with the sample. It means one is going for a qualitative analysis or he is coming for a quantitative analysis. This is, he should know, he or she, the analyst should know. Thus, according to analysis, one should prefer the sample. For example, whether the case is ante-mortem or post-mortem. Urine is the most commonly employed or commonly taken sample for qualitative purpose because majority of the drug and their metabolites of interest can easily be determined and urine can easily be available in ante-mortem cases without invasion of privacy of subject. Moreover, here oral fluids and sweat are being used with increasing frequency. Sample used for the quantitative analysis are selected with the intent of correlating the analyte concentration in these samples with the effect produced by the analytes. As we know that blood is the simple most sample and commonly selected for this purpose because due to its availability and the data of research is in a large number. Now, the other condition that forensic toxicologists should keep in mind is that whether the case is ante-mortem or post-mortem. As we know that in ante-mortem cases, one would have limited sample as only a small number of sample type is available from the living, that is blood, saliva, sweat, urine, hair, vomit, maybe exhaled, unwashed cloth, soil, empty container of the poison and oral fluid. In case of post-mortem, number of samples can be available for analysis such as viscera or tissues. This is most important, which can be generally be obtained as desired from the dead body along with the ante-mortem sampling. But in ante-mortem cases, only ante-mortem samples will be available and collected. So now we are going to discuss the number of samples for the analysis. There is no hard and fast rule that defines the what should be the number of samples. It depends on the analysis and it also depends on the poison. So one thing that should be remembered by the forensic toxicologist is that one should collect multiple samples for analysis. This doesn't matter that the type of case you are dealing, whether it's an ante-mortem and post-mortem. So now I think you are clear with this sample, and with this we conclude module 3. Now we start with our new module, module 4, sample collection. My dear student, sample collection is another important step in sample handling. The collection of samples is the first step in sample preservation and therefore must be conducted with the goal of maintaining the structural integrity of the sample and the concentration of any analyte that may be present, or at a minimum preventive meaningful changes in either, so that false positive or false negative results are less likely to occur or we stop. For this reason, great care must be taken to ensure that to the extent possible, the sample collected are not contaminated with other samples or other substances from the collection implements, and the physical nature of the sample is not altered in a manner that causes the concentration of any drug present in it to be altered. The manner in which samples are collected must be conducted So, there is no question as to the identity of the sample collected. Generally, identification is not a problem since the sample is collected from a known anatomical location and is easily identifiable. With this, we come to this informative module 4's end. Now we are going to start with our new module, module 5, sample container, collection site and sample quantity. So, my dear student, now we are going to learn about the sample container. Sample container means where you are going to keep the sample, collection site and quantity of the sample. So, first of all, we will be talking about the sample containers. Sample container also plays a significant role in the analysis because the concentration of the post-mortem samples varies from one case to another. For example, suppose that we are going to collect blood. You cannot collect it in the beaker. It doesn't make any sense. And you collect the blood in beaker and send it. People will say, what the hell you are doing? So, while collecting the sample, forensic toxicologist has to keep in mind that choosing the proper container according to the exhibit. Otherwise, all the efforts would be worthless. So, let us discuss in brief about some container which are frequently used for the collection. Glass means glass, it is not glass, it is a glass container, all the containers made up of glass. It is frequently used in the collection of post-mortem samples. Thus, glass has some good characteristic like it is inert, it does not contain any plasticizer, contaminant and maximizes storage space. You might have heard about the name Borosil glass, use that. There is only one thing that is arguable with the glass is that possible of breakage. Now we move on to another type of containers. They are the plastic which is now commonly used. So, most type of plastic container are suitable for the collection of solid tissue specimen and gastric contents. The nature of the solid tissue reduces direct contact with the plastic container. Got my point? And the relative amount of drug present in the gastric content will minimize the influence of plasticizers interference. So, my dear student, now we will be discussing about the collection site. Collection site is also one of the factors that affect the concentration of the analyte in the matrix. There is no hard and fast rule for deciding the collection site, but samples should be collected from different part of the body or from different areas of structurally heterogeneous organs such as the heart or the lungs in postmortem. Here I would like to clear one thing, that is collection of samples depends on the condition of the victim. Condition of victim means whether that person is alive or that person is unfortunately died or dead. Moreover, it also depends on other factors such as the collection of blood from fire victims should be collected from internal vessels or the heart because blood collected from the body wounds or cavities may be contaminated with carbon monoxide and cyanide may get absorbed from the atmosphere or the site of the fire. Now we are going to learn about the sample quantity. As we know that for the analysis, a sufficient quantity of material must be collected so that all necessary screening tests, confirmatory analysis and repeat analysis may be performed. So, collection of material depends on many factors such as if the body is putrefied, mummified, skeletonized, and other thing is that body is in ante-mortem condition or in post-mortem condition. If it is in post-mortem, as we have discussed. Moreover, the quantity of the material required also depends upon the number of analyses to be conducted and the detection limits of the analytical methods employed. Generally, we should have approximately 25 to 100 ml of blood, 50 to 100 gram of other tissues and all available bile, stomach content, urine, vitreous humor. If urine cannot be obtained at the time of autopsy from an apparently empty bladder, the bladder should be rinsed or washed with a small amount of water or saline to obtain any residual urine that may be present. So, my dear student, forensic toxicologist can have different conditions. Therefore, according to condition, one has to decide. With this, we come to the end of this interesting module. So, my dear student, now we move on to another interesting module, module 6, sample labeling and sample preservation. So, both sample labeling and sample preservation is an important step for a forensic toxicologist as well as a crime scene investigating officer. So, all the samples, whatever we are going to send for analysis in the forensic science laboratory, that should have a labeling, because it will be good from the examination point of view, because forensic scientist could easily identify what it has or one has sent in the respective department or from the chain of custody point of view. Thus, all samples that are collected should be labeled with a case number and or maybe whichever is the subject name or the person's name, victim's name, sample type, a specific site from which the sample was collected, the name and signature or the initials of the collector, the date and time of collection, and the type of preservative used. Moreover, if labeling is not possible on the container, then it should be written on the tag, and then the tag should be stuck with the container. Moving ahead, we will be discussing about the preservation of the exhibit. Preservation of the storage for the storage purpose, not for the storage, is very important, as improper storage of the exhibit may change the analyte's nature, as well as peculiar characteristics of the analyte may be disappeared. So, forensic toxicologists should be conscious while preserving the sample. Moreover, they should use the protocol for the preservation of the samples. In addition to this, they should preserve the sample according to their nature. This is very, very important. Here we will have some common methods of sample and analyte preservation, such as by decreasing the temperature, that means refrigerated, enzyme inhibitors, alteration of pH, preparation of dried sample. Here we will have or not have detailed discussion on these common methods. So, when you move on to your next semester or when you have advanced or detailed studies, then you will, but I am giving you a brief idea. So, this is it. So, now I think with all these things in these 6 modules, you know how to collect the sample, what are the important samples and all things. So, I think it is very much clear. With this, we come to the end of module 6. My dear student, now we move on to our final module. As you know, well versed with these things. It is module 7 and conclusion. So, as we know that sample handling is the most crucial part of the analysis. As we have talked about sample handling in systematic and step-by-step procedure that contain collection, labeling, preservation and forwarding of the exhibit. If your sample handling is not proper or your instrument is not working properly, indeed, you will not have a good result. Okay, but if your things are perfect, if you are doing it perfectly, your instruments are working correctly, then you will have a best result. No one can challenge it. With all these information, here we are coming to the end of today's lecture. Do keep in mind what we have discussed today. It is very, very, very important. I will be back with more interesting lecture in this series to enhance your knowledge and to work with you more. This is Professor Devashish Bose signing off for today. And if you want to enhance your knowledge and to know more about these kind of things, please log on to our website at www.cec.nic.in for MCQs, quizzes, LORs, etc.